reconciliation and equitable partnerships, and to never cease having immense gratitude for the privilege to work and study and call this place our home. So with that, I am honored to introduce our speaker here, Dr. Nick Ogden, flying in all the way from Montreal. Dr. Nick Ogden is a UK-trained veterinarian who obtained a doctorate in philosophy in Lyme disease ecology at the University of Oxford and studied tick-borne diseases in Europe and Africa as a professor at the Faculty of Veterinary Science, University of Liverpool. After moving to Canada and receiving postdoctoral training in disease modeling at Université de Montréal, he continued research on the ecology of Lyme disease and other zoonoses and climate change as the research scientist at the Public Health Agency of Canada. As a director of divisions in PHAC programs, he directed programs on climate change and infectious disease and community adaptation, and on national coordination, surveillance, and prevention, including Lyme disease and West Nile virus. He is now Senior Research Scientist and Director of Public Health Risk Sciences Division within the National Microbiology Laboratory of PHAC, normally focusing on assessing disease risk by study of the ecology, epidemiology, and generic diversity of vectors and zoonotic and vector-borne microorganisms, assessing impacts of climate change on zoonoses and vector-borne diseases, and developing tools for public health adaptation. He recently led PHAC's COVID-19 modeling and emerging science teams in response to the COVID-19 pandemic. I will pass it over to you.
Okay, thank you. Can you hear me? Yeah, okay, I might be a bit croaky, bit of a kind of post-flu experience at the moment. So thanks for inviting me to speak to you today. I'm going to talk about modeling because we've been through the kind of golden age of modeling for infectious diseases. And during the COVID period, we've been modeling infectious diseases for quite some time, but this is the first time that kind of people knew and that senior managers started to use it. So I will go through a little bit about what we did in the COVID-19 epidemic, but also our more general thoughts, because as I say, we've been doing it for quite a long time. We have a joint research group with the University of Montreal on zoonoses and public health, which is now over 20 years old. And the basis of that was understanding the ecology and epidemiology and synthesizing that knowledge with modeling, spatiotemporal modeling. So we've got a bit of previous on this. Just thought I'd start off about emerging infectious diseases. So diseases emerge and re-emerge by introduction from afar, like West Nile did, and it suddenly kind of finds that it can actually be transmitted. Same thing, I guess, with COVID-19. Geographic spread from one area to another, which is what we're seeing with Lyme disease in Canada right now from the US. Then there's the sort of ecological changes that determine spillover from usually wildlife reservoirs into humans. And that may be two steps. It may be that there is some change in the ecology of transmission of these pathogens, or it may be that we change somehow our contact rates with those the natural reservoir. And again, COVID-19 is an example of that. And then there's emergence because of evolution of pathogens, which to some extent we're seeing with H5N1 across the country. Importantly, again, for those in a biology environment, that this is entirely relevant to you, because most of these emerging infectious diseases are zoonoses. They're diseases, pathogens transmissible from animals to humans, most of them coming from wildlife originally, maybe stepping through to livestock and then getting to people, as maybe we'll see with H5N1. But the determinants of those pathogens is really the ecology of these infectious diseases. There are different types of zoonoses. There are like West Nile, which goes from birds to mosquitoes to birds, and then occasionally to people, but people can't transmit it. Then there's the kind of MERS, CoV, Middle Eastern Respiratory Syndrome, coronavirus, and to some extent, there are Crimean Congo hemorrhagic fever, for example, where people acquire it from an animal host, and then there might be some transmission human to human, but it's not efficient enough for it to persist. And then we have those that are already transmissible. We just haven't encountered them, or we don't encounter them frequently. And that's what we found with COVID-19, right, or SARS-CoV-2, is that immediately it was contacted, whichever that route actually might be, from a bat or an intermediary, but it was immediately transmissible human to human. It's the same with Ebola. And then there is the possibility that if we allow exposure to happen, that one virus will, that a virus that is not well transmissible will adapt to become more and more transmissible. And then we saw that through the COVID-19 thing up to Omicron, which is sort of, you know, almost an order of magnitude more transmissible than the wild type. Behind that, this whole landscape of disease emergence is the issues of the Anthropocene, the climate change, biodiversity change, socioeconomic change globally, interconnected world, which is favoring the spillover of pathogens into the human population and then global spread. And because of that, we recognize it. And as I mentioned, this is a One Health issue, which means that we have to understand really the big picture about what is driving the emergence and re-emergence of these pathogens. We've been working on this for 20 years with our collaborators. Climate change is a key driver at the moment, of course, because it's happening so fast. So warming, Canada is warming faster than anywhere else on the planet. We're going to get changes in rainfall, and we're going to get climate variability and extreme weather events, and obviously a lot of forest fires. So that's just the kind of biological background to it. So from our perspective in public health, what decisions need to be made? And there are two kind of themes to what we do. Of course, we have to do the outbreak management. So when stuff happens, we have to respond. You know, disease emerges, then we have to be able to diagnose it, then respond to an epidemic and put in place surveillance and control and so on, kind of on the fly. Obviously, we want to try and be more prepared, and particularly in the context of climate change, when we have got a little bit of lead-in time, we can do that. I'll talk a little bit more about that. In other words, you're doing sort of model-based risk assessments to understand where and when bugs may emerge in the future, and put in place some kind of surveillance or early warning system so you're a bit more ready for it. So we'll talk about modeling in crisis time first, which is what we've just been through. And the type of modeling that we've been doing is importation modeling. Talk a little bit more about that, about where and when, how many cases will be arriving into Canada. First of all, a key question is, how bad is this going to be? Which is, you know, what is the magnitude of this in terms of how many people are going to get it, how many people might go to hospital, how many people might need ICU, how many people might die. Identifying at-risk populations if we can, in terms of geography or population vulnerability. Assessing the effectiveness of intervention types and designs. And we also have this added value kind of tracking using modeling tools to track the epidemic. Obviously, things like the instantaneous reproduction number, RT, which, you know, the idea that, say, the golden age of modeling is that we have Dr. Tam standing up in April 2020, explaining the basic reproduction number to the Canadian public, which was probably a bit of a first. So the type of mechanistic modeling that we're doing is, I think at least a number of people will be familiar, is the kind of compartmental mathematical mechanistic modeling of the disease transmission. And the population is divided into these boxes of people who are susceptible, people who are exposed, people that are infected and infectious, and then people who have recovered or died. And with the more complex the dynamic of the infection, and the more complex the questions that we're asking, then the more complex the models become. And we had a number of models that we developed internally, pretty much starting off on January 2020. We had, in my team, no experience of modeling infectious, respiratory infectious diseases before that, but we had to get up to speed pretty quickly. So we had the models of varying complexity up to the agent-based type models, individual based models, which are really modeling a more societal approach, which is probably easier for us to use to understand how, closures affect transmission. The model parameters are informed by the literature. We had, and I think it's an important thing here. This is not a it is a mathematical exercise, but it's a mathematical exercise based strongly on the biology of the system. So it's searching the literature. We have a knowledge synthesis team in our division and they were collecting, identifying every publication every day and synthesizing that information to provide the best estimates we have for those key parameters that you need to make a model, like how long is it from infection until someone starts to become infectious? How long before they become symptomatic? What proportion of people actually become symptomatic? How long is the infectious period? What is the proportion of people who suffer severe outcomes according to different age groups and so on? What is the effectiveness of vaccines? What is the effectiveness of interventions? Some things, though, you can't get because there's no data on it, and you have to do that by model fitting. In other words, you're seeking particular key variables by a number of different fitting the models to data. In other words, getting a plausible range of parameter values for missing data on the basis of the surveillance information that you have. So this is the first objective. This comes out of Dr. Tam's presentation in April 2020, a stylized version of the simple modeling, which is a sort of, oh my God, here is a COVID, here's a bug, which we've no immunity as far as we're in the population. We have no antivirals and we have no vaccines. And somewhere between, before it comes to an end, somewhere between 70 to 80% of the population, maybe a bit less, are going to have to be infected. And the case fatality or infection fatality rate is about 1%. So about 1 in every 100 people infected will die. And then we sort of identified that we are going to have to do something about this because of that. It is a real difficult problem. The second objective we had, having identified that, well, right at the start, we're going to have to shut down in order to stop transmission. And we're going to discuss this afternoon about who does what. Like, we are advisors. The people who make the decisions about shutdowns are governments, provincial governments, by and large. So we have no kind of, we provide the evidence. In other words, it's going to be bad. And the only thing that we can do right now, before we and we had to build up the whole surveillance system, and the initial shutdown was a pause to allow that to happen. But then we were asked, well, okay, this lockdowns thing is really bad for people. economies, everything. Nobody wants it. So there are other public health measures that we can use. Test and trace, identifying, in other words, instead of focusing the interventions on everybody, you're focusing on just those who are most likely to be infected. So you want to identify the people who are infected and then do contact tracing to identify who they've been in contact with and put them in quarantine, and then perhaps with other measures like distancing, physical distancing. So when people meet, the probability of transmission is reduced. That maybe if we use those together, we can actually open up without having to keep people all locked up. So we undertook this this modeling, and the outcomes are that, you know, if it works well, you'll get the graph on the left. The green bar here is the lockdown period. The black line is the first wave, and of course some stochasticity in the model. And you can see if we do the job right, lift restrictions, and we've got enough test and trace and enough effectiveness of distancing and so on, it maintains under control. But as we can see on the right, this is the thing that a lot of people didn't get, is that we're still all naive. Very few people acquired infection in the first wave, and if you just let things go, well, it'll just rip off, and again, 70 or 80% of the population are infected. So that's what you can see on the right, and of course, as we know, that's what happened. Of course, we lifted the restrictions, and one of the reasons, and I'm not going to go into details about this, is that we looked at what is the proportion of cases and contacts that have to be detected in order to control transmission. And it was a high percentage, right? Which is really difficult. Not only a high percentage, not shown here, it has to happen really quickly. And when there was not enough reagent to do PCR testing, and when there's not enough people in the public health system to do it, because in my opinion, they've been underfunded for a long, long time. When there's asymptomatic and pre-symptomatic transmission, it makes this method of control exceedingly difficult. So then we realized that governments are going to have to, or they're not able to control the transmission with these alternative methods. So how can we use combinations of these efforts, and particularly what are those? How to use lockdowns? Like, are there things that you can open? What are the risky things? And of course, the risky things are the workplaces, mixed age venues, and so on. The kind of leisure areas were the most risky. And then we looked at how's the frequency. If you really have to do lockdown, how do you do that? And the upshot is that you have to do it. Ideally, you would do it early, and you would do it long enough to get the transmission down to a low level that at least distancing and contact tracing can be having an impact. It may take off again, but at least you're keeping the lockdown for as short a period as possible. On the right, see the work. On the left is work by Vicky Ng in our team. On the right, work by Amy Herford. And what is it, Amy? Don't hesitate, re-escalate was the answer. That if you have to, if you first see things taking off, well, get in there early and do a circuit breaker shutdown. But that's not what happened, unfortunately. Importation modeling. This is a network of the network model of the all of the international air transport network, with behind that values obtained for prevalence of infection and prevalence of different variants, travel volumes, and so on, in order to estimate as best we can what are the number of cases that are arriving in Canada, from where are they coming from, and what are the variants that are coming in. So we had this done every week from throughout the pandemic. And it was also border crossing from the US as well. And these are the sort of early warning analysis and forecasting sort of things. So looking at now, or recent past rather, is the RT, short range statistical model, what's going to happen next week. The long range dynamic modeling, initially done by Simon Fraser, Carolyn Colin for us, but then in collaboration with McMaster, we developed our own kind of model. And for the long range forecast, which some of you will have seen and get in Dr. Tam's presentations. And then more towards the end, using wastewater for estimating RT and producing a forecast. We were asked to, when the vaccines appeared, thank goodness, what percentage of the population has to be vaccinated for us to lift any restrictions. So we were tasked with that. Initially, we didn't know how the vaccines were performing, so we had to have loads of scenarios of what would happen if we had different percentages of coverage of 1 and 2 doses, different performance of the vaccines against infection and severe outcomes, and then come up with kind of like, according to when we got more information about how the vaccines were performing, we will be able to be more certain about when we can, what coverage of the population we could start to open. And you may remember the Prime Minister mentioning the one-shot summer, two-shot fall thing, which was based on our initial modeling. So after a two-shot fall, we can lift a lot of restrictions. But they all knew that there's uncertainties about that. And one of them was having just seen this alpha variant appear, which was more transmissible and more virulent, in other words, caused more severe outcomes of infection. But if we get another that's even more transmissible and more virulent, we're going to have to have an even higher proportion of the population vaccinated. And of course, that's what happened when we had on the left without Delta, on the right with Delta. So in other words, and the red line you see there these are hospitalized cases from our agent-based model, and the red line is our kind of average across Canada hospital capacity. And you can see that with Delta, that one-shot summer, two-shot fall, there was a risk that we would bust our hospital capacity. So that meant that we had to have a bigger control. Unless we did the counterfactuals, what would happen? We've heard that there's been a lot of negativity about modeling. So it's been a golden age of modeling, but there's been a heck of a lot of negative. Oh, it's a load of rubbish. Oh, it never happened. Oh, it was never as severe as those modelers were just scaring us. And there was an awful lot of that. And so we did this counterfactual modeling study to try and describe what happened and what would have happened if things had not come to pass as they did in Canada. So that's the golden age, but peacetime, what are we doing in peacetime? Again, we're doing, connect camera. Okay. We have previously used the word quantitative risk assessment because people, when I talk to a senior manager before COVID and I say, I'm doing modeling, they'll go, what's that? And I go, well, we're producing a quantitative risk. It's our risk assessment. I get that. So now everyone knows what modeling is and everyone wants modeling because they understand how it works. But we've used a lot of risk assessment words in the modeling world. So a lot of it is about pre-detection of things that are going to happen. As I mentioned, it's trying to identify where emerging bugs may arrive in Canada and when they might arrive in the future, particularly with climate change. We're doing estimates of importation, which we do all the time now, because if there's an Ebola outbreak in Africa, we go, how many people are coming to Canada? How many cases? How many people might appear here? Understanding risk in terms of geography and vulnerability, of course. Assessing effectiveness of intervention types, which is what we're doing all the time. I'll talk a little bit more about that. And we're now doing the forecasting of endemic diseases, so respiratory infectious diseases particularly. So climate change adaptation, I'm not going to go through this in detail, but this identifying what the risks are, putting in place surveillance to actually detect when these things emerge, and then developing tools for public health to be ready to respond. And I'll talk a little bit more about each of those. So again, it's a One Health thing. The first is using our mathematical models, which may be understanding the dynamics of transmission of a tick-borne agent like Borrelia burgdorferi, the agent of Lyme disease, a mosquito-borne disease like West Nile, or Eastern equine encephalitis, which has arrived from the US and into Canada recently, or lacrosse virus, which is just across the border and may come across to us. If we have enough knowledge to develop of the ecology of these diseases, then we can develop mechanistic models, which we can use to synthesize all the information about the environmental and other determinants, including the climatic ones. In other words, what happens if it gets warmer versus what happens if we cut woodland down and replace it by farmland. This is an example which I started back in 2003. We fortunately had a load of information about the life cycle of the tick, Ixodes scapularis, and courtesy of Robin Lindsay's thesis, who's a colleague who's at NML Winnipeg, who's now retired. But he basically did it so that we could understand what all of these, what's the mortality rates of these ticks, what's the development rate of the ticks, how does that depend on temperature, and so on. And that allowed us to produce simulations to identify what is the climatic limits of the tick. In other words, and I should say that it's not because it's too cold in winter, because as long as the habitat's good, the ticks survive perfectly well. Maybe minus 40 above, but it's like minus 3, 4 in the duff layer, and the ticks have got a bit of antifreeze, and they survive perfectly well. What climate change is doing to the ticks is, or what climate is doing, is that it's the duration of the life cycle. It's the effect of temperature on how fast one stage develops to the next. So and they've got a pretty constant per capita daily mortality rate. So with a particular climate, the length of the life cycle is this. If it's too cold, the life cycle is this, and the probability that the larva here survives to be the adult here, if it's below 1, well, the reproduction number for the tick is below 1. However, the warmer it is, the shorter that life cycle it becomes, and the more likely that that immature tick will survive to be a reproducing adult. And that is kind of the basis of this. And we've done a number of studies using, well, we know what the climatic limits are, so therefore we can use climate models to say, well, what's going to happen in the future. And then we validated it with an extensive amount of field study, and which has shown that it validates it really well, and that it actually has allowed us with other surveillance data to really validate it, that there's pretty confident attribution to recent climate change. In other words, it's the geographic pattern of spread and the temporal pattern of spread has been associated with the changing pattern of temperatures in southern Canada, which are not explained by previously normal changes in climate. So in other words, it all started off at a couple of years after the temperature in southern Canada started to change from from a trajectory or to a trajectory that can only be explained by anthropogenic climate change. But it kind of illustrates what we did. We did the modeling. We identified that we might have areas that Canada already, and in 2007 and 8, we went out and identified that, oh yeah, early signs of tick populations setting up more widely. There were a few populations known beforehand, which meant that we were allowed to put in place surveillance, field surveillance for ticks, surveillance for cases in humans, and information for the doctors about Lyme disease, because they've been told forever and a day that we don't have it in Canada. And now they had to change that idea, so we had to educate the doctors that it's on its way. Educate the public or provide the information to the public in order for them to protect themselves as best as we can. Now, of course, it is emerging, but it did allow us to get ahead of the curve and put in place at least some public health programming to do surveillance and prevention. We're doing this with other threats, of course, that are heading our way, including here this is the Lone Star tick. Some of you may have heard about this in the media. This is a tick that it does transmit some unpleasant bugs, Ehrlichia chaffiensis, cause of monocytic ehrlichiosis in people, heartland virus, which occurs in the Midwest of the US. It's not established here yet. It doesn't seem to hitch a ride on birds very easily. I don't know why, but the number of ticks we see in passive surveillance, which are usually ticks carried in on migratory birds, is actually very, very low compared to what we saw with the black-legged tick vector of Lyme disease. But it's the cause of meat allergy due to it produces a sugar in called alpha-gal in the saliva, which sensitizes us to alpha-gal, which is also in red meat. So you can get some quite severe allergic responses if you've been bitten on a number of occasions by this tick. So anyway, it's kind of on its way. Newfoundland not looking greatly at risk until towards the end of the century. So I mentioned that we were modeling spread in Canada as well. And well, I was talking to some of Andrew's team earlier on. And this is the work done by Olivia Tardy, which is an explicit, she's a behaviorist, understanding how the behavior of animal hosts is allowing the actual spread of the tick, so that we can not only just say, all right, here's environmental suitability in these future time slices improve so you can get ticks in these areas in the future. We can actually model more explicitly the spread which is more explicit both in terms of space and time, where and when these ticks and the bugs that they're going to be carrying are going to get there. We're also identifying the risk of things coming from afar that we haven't seen for we really didn't expect to see. So, you know, things like chikungunya and dengue and Zika virus are tropical. But Canada's warming so fast that we're now starting to see the climatic suitability for transmission and also for the vectors that we haven't had before, these Aedes species vectors. So Aedes albopictus, the Asian tiger mosquito, has set up home now, pretty much permanent, well established in the warmest part of Ontario, in the kind of Windsor area. And it will be, at some point, warm enough for transmission to occur. If somebody comes back from vacation and is viremic, then there is a possibility for local transmission. As there was an outbreak of chikungunya in Italy, which was quite extensive, and was quite extensive before anybody had identified, A, that the Aedes albopictus had really set up home, and B, that these summer infectious disease was actually chikungunya and not some other bug. We're modeling climate change impacts on food and water safety as well, how climate change may impact the continuum of contamination of foodstuffs from farm to fork, and also antimicrobial resistance genes as well. Indeed. I mentioned that we're now doing this respiratory disease modeling to forecast. We're piloting this for the first time this year, so we will be doing COVID forecasts. We'll be doing, and these are long-range forecasts, so 6 weeks to 2 months ahead. So COVID, influenza A, we had planned influenza B, but it looks like it's gone, an RSV. And this is work being led by David Champerton in our division. So we talked about the kind of mechanistic modeling, but we also do the kind of pattern match. When we haven't got enough information to build a mechanistic model of any that is meaningful, we can still have a stab at understanding what the ecological niche is by, as you know, a lot of people use the machine learning methods based on the knowledge of species distributions to identify what are the environmental determinants, which and also socioeconomic determinants of the occurrence of bugs. And so we are doing this, and we've done this with, again, with these Aedes species, and then assuming that our ecological niche is accurate, that with a warming climate, what will happen, what parts of Canada may we see these exotic vectors emerging? And I think all of you are probably going, well, there's a whole lot of bugs out there that might come and get us, but and these projects are quite long. So how do you choose what to do this work on? And the answer to that is we use a multi-criteria decision analysis method, which I won't go into now, but it's at least a way of saying that we have considered the attributes of these bugs in terms of the kind of impact they might have if they came to Canada, the extent to which includes like the potential for transmission, pre-existing immunity, the possibility of the virulence of the bug. Is it easily treatable? Do we already have control methods and so on in place? And bringing this together, informed by both expert and stakeholder opinion, we can then put all of that information together and decide, okay, what are our priorities? Which is the #1 bug that we have to go for this year? So in other words, we've got an open, transparent way of saying what we're spending the taxpayers' money on in this regard. Tools, perhaps the the tools for public health, the most considered in the space of climate and weather sensitive diseases is forecasting, weather-based forecasting of infectious diseases. If we're based on human cases, and this is in the context here of West Nile virus, by the time we've got human cases, it's too late, really, to act. It's already happening. The epidemic is upon us. And if we then go out, well, we may curtail the epidemic a little bit, but it's kind of too late once we've got human cases, because there's a lag between not only people acquiring infection, but before they're detected and diagnosed and reported and so on. So we know we do mosquito surveillance, but again, there's a lag in that whole system as well. If we can get ahead of the curve on that and understand what are the climatic or weather determinants of, one, the abundance of vector mosquitoes, and two, the proportion of those mosquitoes that are infected, and we can say we've got an epidemic coming up, particularly in the context of the fact that we're going to get more climate variability and extreme weather events, heavy rainfall, heat, which is going to drive epidemics of mosquito-borne diseases, because they've got most of them got short life cycles. The mosquitoes can respond very quickly to changes in weather. So ideally, we will be doing weather-based forecasting of mosquito-borne diseases. So instead of just saying, putting a poster up in kind of medical clinics about, you know, wear bug spray or whatever at the beginning of the year, and everyone forgets about that, you can actually go out with campaigns, go like, if you didn't do that before, right now, you got to do that. You got to put this, because West Nile outbreak is on its way. And you will hopefully get more buy-in from the public if they're told really when they need to do stuff. That's the objective. We're doing modeling to, and have been for some time, understanding what are the best patterns to control rabies. We kind of forget that rabies is this really unpleasant endemic disease that we control by these huge campaigns of vaccination, and actually making these huge and expensive campaigns most effective is one of our objectives. I did not in there, I thought I missed out one of the products for public health, and that comes from our knowledge synthesis team. So when we've identified that there is a risk for a particular bug, then public health needs to know about this bug. So, and how to control it, how to undertake surveillance for it, how to diagnose it, and so on. And our knowledge synthesis team kind of moves into action to produce the knowledge that we have right now about these bugs, so that public health and medical practitioners are going to be have resources at their fingertips in order to respond to these problems when they arise. So, concluding, COVID-19 is the golden age of modeling. They're in part because they were recognized as being useful and effective. COVID-19 is a poster child for how for the emergence of infectious diseases globally, and we're in a better place. However hideous it was, we're now in a better place to do the modeling work than we've been previously. So, and we now have a kind of good old program on modeling infectious diseases, both endemic and potentially emerging. But it's all about the biology of the system, and so it's a big research project, which includes understanding ecology, epidemiology, understanding the methods we use for modeling, and having people who are ensuring we have people who've got the deep knowledge and specialized knowledge, but also recognizing that One Health approach, that we need people who are specialized in multidisciplinarity to really be able to get that One Health approach. So I'll stop there, take any questions.
Thank you so much. Your team has a lot on your shoulders. Okay, so if anyone has a question, I'll just bring you the microphone so that people online can hear.
Firstly, thanks, Nick, for such a great talk that motivates the importance of modeling for all these public health questions. I think that was a really good illustration of the value of modeling. I wanted to ask about fundamental niche, realized niche, and dispersal limitation, and with respect to maybe missing data or priorities for data collection. So, you talked a bit about the influence of temperature on like the life cycle duration for spatial spread problems, like how quickly is something going to invade. Something else that's going to determine that rate of spread would be dispersal, like for, I guess, individual, like infected, like the hosts and the vectors, how far away from where individuals are born do they end up reproducing? Do you think we need like more data on dispersal and movement to better be able to answer these questions about spatial spread?
Yeah, absolutely. I mean, take the modeling work that Olivia's doing, Olivia Tardy. It's asking questions. It's not necessarily right now saying this is how it's going to be. It asks the question: what is the most important piece of, what are the key determinants in this system, and what do we not know about? What have we got very poor information about? I mean, as I mentioned earlier, she's combining like the sort of pattern matching machine learning species distribution models with spatial spread, mechanistic models, but the data that are underpinning the species distribution models are often incomplete. They're not good. They've got false positives and a whole host of false kind of negatives around the world. So yeah, I mean, it's one of the things that we encounter frequently in this process is understanding what we don't know. And in other words, that inspires us to then go, right, well, we need to fill that gap by doing something prospective. So yeah, I mean, species movement is just one part of that.
Any other questions?
Thanks, Nick. So what do you think? So I'm convinced that for the next thing that comes along, your group is ready for modeling it better. Do you think as a society, Canada or other countries are better prepared for the next thing, or is it just going to be the same all over?
I hope we're going to be better placed in terms of my team. In terms of, are we globally better? I don't think so yet. I think we have still really got to meaningfully explore what went well and what did not go well. What are our significant gaps? And there are a number of ways of looking at that, and that is like being better prepared to respond when something happens, being better able to kind of as a global community to identify when something happens, And also to be, as was brought up, that we have an external community of practice of modelers around Canada. At the meeting on Wednesday, the idea is that how do we make ourselves more resilient to in terms of, we learned that COVID-19 transmitted in places easily when there was poor ventilation. So can we not get better ventilation? And key amongst those bugs that are scary are respiratory infectious diseases, be they influenzas or coronaviruses or whatever. So, maybe doing something about that would be something sort of systematic would be a worthwhile thing to do. But I wouldn't stop there at resilience.
Sure. Short follow-up. So there's some people who are proponents that one way to be prepared for what might come is to go and find all the possible viruses out there in bats. And then maybe when we know that what's there, we'll be more prepared. And that costs a lot of money. And it's controversial because some people think that's the kind of work that leads to the COVID-19 pandemic. So do you have any comments about that? Is that a valuable exercise, or should we not be looking for new viruses in these potential reservoirs?
I understand the fear that if you find something nasty, then someone might grow it and use it for bad purpose. But we've already got, quite a lot of bugs can be used for bad purpose. I'm unconvinced that shutting our eyes and putting our hands over our ears and preventing us having knowledge of what's out there is a good way of doing business.
Thanks for the talk. I'm an ecologist, not an epidemiologist, but I do sit on advisory committees to governments, and I would love to hear a little bit of commentary on you on the challenges of translating science and scientific uncertainty to decision makers. And if we contrast the way Canada reacted to other countries, I feel like we did better than other countries. But what advice would you give to anybody in the room who might find themselves in that situation, and where the government is expecting science maybe to have the answer when we don't, we have uncertainty, and needing to balance that with other competing things like economics and so forth?
Yeah, it is something that it was another of the things we built in flight, right? So I think being, and this is, someone needs to write a book about it, right? For the next pandemic, because this is crucial. Not only how do you communicate this information to senior managers and decision makers who are politicians, but also to the public. And we need to go back and have a look at it. I probably don't have an immediate answer, but I do know that we had senior managers in the agency who got it, who understood it, and understood that the modeling doesn't necessarily need to be precise, as long as it is giving, and then, in other words, the number of people who are going to go to hospital is not 147. It's somewhere in this range. But if we do that, we'll probably get this range. But if we do that, we're going to get probably most likely a smaller range. In other words, we're better to do that. Is something that they get, right? And we're able themselves to then communicate both to the public and to the kind of to politicians. We have to recognize also that the politicians were in place. The health ministers, Paddy Hajdu was a public health person. And then after that, Dr. Jean-Yves Duclos, who's an economist, head of a U Laval fellow of the Royal Society. So these were not people for whom this was alien. So maybe we were lucky in that respect.
I think we have time for a couple more questions.
Thanks. I had a question that is, I think, similar to Yolanda's, which is that as you've indicated as modelers, you guys are giving advice to decision makers, and ultimately decision makers are making a decision, weighing that evidence, perhaps against other evidence. I find often when I'm listening to politicians and others describe why they've made the decision, there's this like, we're following the science. And it seems like a way of being almost like shirking responsibility for the values judgments that are being made based on different types of evidence. And I'm wondering if you could speak about, I guess, your comfort or your assessment with how decisions about public health measures were made based on evidence from multiple places and based on values judgments. I guess, do you feel like the values judgments that people are making are being communicated well, or do you think it's a bit of a shirking of responsibility to just say, oh, we're following the science and that's what it is?
I might have my own opinions about that, but I'm not going to share them.
Fair enough.
It's my job to provide the evidence to our senior managers, to the civil servants, who then provide that information to the politicians, who then take those judgments. That's their call, and that's their job.
Thanks.
Thank you for the presentation. COVID-19 showed that we were more or less well prepared to model things that we know how they work. But what it showed also was that we were not really prepared for things that can be catastrophic in terms of modeling. Do you think now we are better prepared in that sense?
I don't know, but I think you're right. Is that there was my perception at the start was there was no real understanding of how catastrophic this could be, and I think that included me, right? I mean, it's sort of, you know, you do a bit of modeling, 70-80% of the population. You know, just short of 10% or 1% of the population dead. And you go, well, can't be right. Go back and have a look at that again, you know. And so there was this sort of like, and I remember that moment of, yeah, that's right. There was a pretty hollow feeling. But so we weren't ready for that. I think, will we be ready for the next one? Will we have forgotten? I don't know. Maybe.
My other is more like a comment than a question. We live in a privileged place. We live in a great country. But preparedness and modeling, and it is a global thing. And I think it's a great disparity in the knowledge and the capabilities and the skills to model in other places than most places of the world.
Sorry, there is a disparity here
in the preparation and the skills to predict, and I think something has to be done about it, because the next pandemic will not start in Canada.
Yeah, no, it's absolutely true, and that is the. But it's not just modeling that is part of this whole international community of haves and have-nots. I mean, it's in the disease context, it was everything, right? It was surveillance, it was rollout of vaccines, it was knowledge, risk perception. In some countries in Africa, somebody did a study looking at what people's perception was, and when they've got a relatively short life expectancy certainly compared to us, and so many challenges that please take time off to go and get a vaccine. Their time scale is a week, and because that's what their circumstance demands of them, that they have to concentrate on the next week, not what might happen some months down the road, which is just an illustration, I think, of the challenges.
All right, I think we're a little bit past 1. I don't think there's any questions online, so, but yeah, thank you so much, Dr. Ogden. And thank you so much for everyone who came out. It's nice to see such a good turnout. There's lots of snacks and coffee. Help yourself.